Vitamin D, long celebrated for its role in bone health and calcium regulation, has emerged in recent decades as a critical modulator of immune function, with a particularly important role in activating autophagy within cells of the innate immune system. Far beyond its classical endocrine functions, vitamin D exerts profound effects on cellular defense mechanisms, enabling the body to respond to microbial threats with remarkable precision and efficiency. Among its most fascinating biological actions is the enhancement of autophagy in immune cells, especially macrophages, which serve as frontline defenders against pathogens. Autophagy, often described as the cell's self-eating process, is not only a tool for cellular renewal and detoxification but also an essential component of antimicrobial defense. In immune cells, autophagy plays a key role in the identification, encapsulation, and degradation of invading pathogens such as bacteria and viruses. This form of targeted degradation is known as xenophagy, a specialized type of autophagy that helps eliminate intracellular infections. Vitamin D supports this process at multiple levels, making it indispensable for a strong, adaptive innate immune response. At the molecular level, vitamin D activates autophagy in macrophages by upregulating genes involved in pathogen recognition and response. When vitamin D binds to its receptor, VDR, in immune cells, it triggers transcriptional activation of key antimicrobial and autophagy-related genes, such as cathelicidin, LL37, an antimicrobial peptide that disrupts bacterial membranes, and Beclin-1, a central regulator of autophagosome formation. These genes enhance the immune cell's ability to engulf and degrade microbial invaders while simultaneously reducing inflammatory overload. This is crucial because a properly regulated immune response requires both potent attack mechanisms and controlled resolution. Autophagy provides both the cleanup function and the modulation of excessive inflammation. One of the most well-studied models of vitamin D-induced autophagy is in the context of tuberculosis, TB, a bacterial infection that survives by hiding inside macrophages. Research has shown that when macrophages are treated with vitamin D, they become far more effective at inducing autophagy and killing the tuberculosis pathogen, Mycobacterium tuberculosis. This effect has been linked to the upregulation of cathelicidin and the restoration of phagosomal acidification, key steps in the autophagic destruction of intracellular bacteria. Such findings underscore the broader principle that adequate vitamin D status is essential not only for infection resistance, but also for effective immune cell cleanup and resolution of inflammation. Vitamin D's role in enhancing immune autophagy also has implications for viral infections. Some studies suggest that vitamin D may aid in the autophagic response to certain viruses by improving antigen processing and limiting viral replication. In addition, it may help modulate the cytokine response during viral infections, reducing the likelihood of excessive inflammation or cytokine storms, phenomena that can occur when autophagy fails to regulate the immune response effectively. Beyond infectious disease, vitamin D-induced immune autophagy may play a role in autoimmune regulation. By promoting autophagy in dendritic cells and macrophages, vitamin D helps maintain a more tolerogenic immune profile, which could reduce the likelihood of the immune system mistakenly attacking the body's own tissues. This has led to interest in the use of vitamin D as a supportive agent in autoimmune conditions like multiple sclerosis and rheumatoid arthritis, where dysregulated autophagy and immune function intersect. From a nutritional and lifestyle standpoint, supporting optimal vitamin D levels is both accessible and highly beneficial. The most natural source is sunlight, particularly UVB exposure, which converts 7-dehydrocholesterol in the skin to pre-vitamin D3. Just 10 to 30 minutes of midday sun exposure a few times per week, depending on skin tone, latitude, and season, can be enough to maintain adequate levels in many individuals. For those with limited sun exposure, vitamin D3 supplements offer a reliable alternative, especially during winter months or in populations at risk for deficiency. Fortified foods such as dairy products, plant-based milks, and certain cereals can also contribute modestly to intake, though they may not be sufficient on their own. Because vitamin D is fat-soluble, it is best absorbed when taken with a meal containing healthy fats, such as avocado, olive oil, nuts, or fatty fish. Maintaining sufficient magnesium levels also supports the conversion of vitamin D into its active form and enhances its immune benefits. When combined with other autophagy-supporting nutrients, such as omega-3 fatty acids, polyphenol, or fasting protocols, vitamin D can synergistically enhance immune resilience and cellular regeneration. In conclusion, vitamin D plays a vital role in enhancing immune-related autophagy, particularly in cells like macrophages that rely on this process to fight infections and clear debris. By upregulating antimicrobial peptides and autophagy genes, vitamin D transforms immune cells into more effective and self-regulating agents of defense. This not only protects against bacterial and viral infections, but also supports broader immune balance, potentially reducing the risk of chronic inflammation and autoimmunity. With its dual accessibility through sunlight and supplementation, vitamin D offers one of the most practical and powerful tools for supporting innate immunity and promoting healthy autophagic function throughout the body.